Dear students, I am Dr. Y.S. Rajput, Emeritus Scientist, working at Animal Biochemistry Division, National Dairy Research Institute, Karnal. Welcome to EPG Pathshala. Today, I will be teaching you one of the very important technique of isoelectric focusing. Isoelectric focusing, if we see, this is a technique which is used for separating two molecules which differ in their isoelectric point as low as 0.01 pH unit. This technique is basically used for identification of unknown protein molecules by measuring their isoelectric point. Other biochemical techniques, these can be used for identification as well as the quantification of protein molecules. Amongst them, we see the electrophoretic techniques, which are largely used for the identification of the molecules in some situations like agar-gel electrophoresis and SDS-based electrophoresis. These can at times be used for the separation of the molecules for the subsequent use. If we see the chromatographic techniques, there the main purpose is to separate the molecules. So for separation purposes, chromatographic techniques are best suited. But at the same time, these can also be used for the quantification of the molecules. I will be covering in this presentation theoretical as well as practical aspects of isoelectric focusing. I am sure that after listening to this lecture, you will be able to perform your experiments more carefully with a success rate as much as high as 100%. The objective of presentation is to make you understand the principle of isoelectric focusing. I will also be covering how the technique of isoelectric focusing is performed. The technique of SDS space and isoelectric focusing can be used in combination for enhancing resolving power, and this is referred as two-dimensional gel electrophoresis. This aspect will also be covered. It is important to understand what contributes to charge on protein. Proteins are polymers of amino acids which are linked together by peptide bond. Amino acids contain amino group as well as carboxylic group. Carboxylic group of one amino acid is linked with amino group of other amino acid, and thus these groups are involved in peptide bond formation and will not contribute to charge. However, alpha amino group of first amino acid and alpha carboxylic group of last amino acid in protein or peptide will contribute to charge on molecule. Different amino acids contain different side chains, and some of them do contain groups which can contribute to charge on protein. For example, side chains of aspartic acid and glutamic acid have carboxylic groups, while that of lysine has amino group. Similarly, imidazole group of histidine, guanidino group in arginine, hydroxyl group in serine and threonine can contribute to charge. Thus, pK of alpha amino, epsilon amino, alpha carboxylic, beta carboxylic, gamma carboxylic, guanidine, hydroxyl, imidazole groups, and number of these groups in protein, and further pH will determine the net charge on protein. Dissociation of carboxylic group will lead to one negative charge, while dissociation of amino group will abolish one positive charge. pH and pK decide extent of dissociation of groups, and extent of dissociation will decide charge contribution to molecule. Extent of dissociation can be determined by use of Henderson-Hasselbalch equation. When pH is equal to pK, there will be 50% dissociation. When pH is 1 unit higher than pK, there will be about 90% dissociation. When pH is 2 units higher than pK, there will be about 99% dissociation. When pH is 1 unit lower than the pK, there will be about 10% dissociation. When pH is 2 units lower than the pK, there will be about 1% dissociation. Isoelectric point of protein is pH, at which net charge on protein is 0. And at this pH, protein in electric field will not migrate. At pH value higher than the Pi, the protein will have net negative charge and move towards anode. However, at pH values lower than Pi, protein will have net positive charge and move towards cathode. Most of proteins have PIs in the range of 4 to 7, and therefore at alkaline pH such proteins will have negative charge, while at pH lower than 4.0 the net charge will be positive. Consider the situation where pH is increased from say 2 to 10. At pH 2, protein will have net positive charge. The net positive charge will gradually decrease with gradual increase in pH, and then charge will reach 0. Further increase in pH will result in net negative charge, which will continue to increase with increase in pH. Also, it is made clear that net charge on protein will continue to increase as one moves away from its Pi values. Isoelectric focusing is an excellent electrophoresis technique for the separation of amphoteric molecules such as proteins. Separation in isoelectric focusing is possible only when proteins differ in their isoelectric points. The resolution of technique is very high, and two proteins differing in their Pi values by even 0.01 pH unit can be separated. In electrophoresis, buffer is used to keep charge on protein constant during migration. In isoelectric focusing, buffer is not used. So this should be noted very carefully that we do not use any buffer in isoelectric focusing. Proteins move freely through the pores of support, generally of low concentration of acrylamide in the range of 5%. For high molecular weight proteins, agarose gel can also be used. Linear pH gradient is prepared in support. There are two approaches which are used for preparation of pH gradient in gel. In first approach, Ampholytes are used, while in other approach, acrylamido buffers are used. Anode is in contact with 0.1 to 1 molar phosphoric acid, while cathode is exposed with 0.1 to 1 molar sodium hydroxide. End of support gel closer to the anode will be low in pH, while gel at the other end will be at higher pH. Thus pH gradient is formed, which linearly increases from anode to cathode. Consider the situation when protein is loaded at a defined point in pH gradient gel and electric current is applied. Because of the pH gradient, pH of gel will differ at every location. pH at the point of protein loading and its Pi will decide the direction of migration of protein. If pH is higher than Pi of protein or protein is loaded closer to cathode. Protein will be negatively charged and will move towards anode. While it is moving towards anode, protein will come in the vicinity of gel having lower pH. At this decreased pH, net negative charge on protein will decrease and this will result in lower migration rate. Thus, in pH gradient, the rate of migration of protein will continuously decrease and protein will not move further when net charge on the protein becomes 0. Also, consider the situation where same protein is loaded closer to anode. At low pH, protein will have the positive charge and it will move towards cathode. Initially, protein will migrate at a faster rate because more net positive charge and protein will continuously come in contact with gel having higher pH. This will result in decrease in net positive charge and consequently migration rate will decrease. When pH is equal to Pi, protein will not move. Now it should be clear that protein sample can be loaded on either end of gel. In isoelectric focusing, protein gets accumulated at its isoelectric point. Ampholytes are mixtures of polyamino polycarboxylic acids and are commercially available as formaldehyde or biolite. These are also referred as carrier ampholytes or amphoteric electrolytes. Multiple carboxylic and amino groups are present in ampholytes. Molecular weight is in the range of 300 to 1000 Daltons. Pi values of these molecules is very close to each other, and these molecules also possess high conductivity. Depending upon their Pi values, these molecules will move in electric field and finally fixed in gel as per their Pi values. Polyamino polycarboxylic acids have Pi values corresponding to pH gradient. Ampholytes with control number of amino and carboxylic groups can be synthesized. Ampholytes are very expensive. Only few companies are marketing this product. Ampholytes are available for different pH range. Broad pH range and narrow pH range ampholytes are available. The separation of protein on broad range ampholytes will provide information on approximate Pi values of proteins. Exact Pi can be determined when narrow range pH gradient ampholytes are used. Slope of pH gradient depends on pH range of ampholytes and distance between electrodes. pH gradient from ampholytes can be easily prepared in the laboratory. Ampholytes are added at a concentration of 2%. Ampholytes at a concentration of lower than 1% provide unstable pH gradient. Ampholytes above 3% are difficult to remove from gel and remaining ampholytes interfere in staining process. Earlier, 1 to 2 millimeter thick gel was used in isoelectric focusing. Nowadays, ultra thin gel, that is something around less than 0.5 millimeter, can also be prepared. These gels can be casted between glass plates separated by spacer, like in preparation of SDS paste gel. Isoelectric focusing gel is polymerized through photochemical initiation of mixture containing acrylamide, bisacrylamide, riboflavin, ammonium persulfate and tamide. Acrylamide concentration is usually kept around 5%. urea and or non-ionic detergent such as CHEPS, NP40, Triton X100 can also be added in need-based situations. Urea disrupts hydrogen bonding, which otherwise can lead to aggregation of molecules and affects its mobility. Membranes proteins require detergent for their solubilization. Sodium dodecyl sulfate cannot be used as it will change the charge. Photochemical polymerization requires exposing plate to fluorescent lamp. Acryl amido buffers contain both reactive bonds as well as buffering group. These are derivatives of acrylamide and contain reactive carbon-carbon double bond and carboxyl or tertiary amino group. Carbon-carbon double bond can take part in gel formation, hence acryl amido buffers are incorporated in gel. Here pH gradient is immobilized in gel. Acryl amido buffers of different pK values are available. Preparation of pH gradient requires linear mixing of two acryl amido buffers. Narrow as well as wide range pH gradients in the range from pH 2.5 to pH 11 can be created. Preformed thin isoelectric focusing gels are now commercially available for different range of pH values. Isoelectric focusing can be performed by using immobilized pH gradient strips, popularly referred as IPG strips, available in the market. IPG strips are very simple to use. These are stable even during extended running of isoelectric focusing. Generally, broad range strips are preferred for resolution of protein. PI values of maximum proteins are in the range of 3 to 10. Commercially available IPG strips are in dehydrated form. These are rehydrated by using the solution containing deuterionic detergent such as Triton X100, dithydratol as a reducing agent, Ampholyte solution as a desired range, and a trace amount of bromophenol blue. Sample can be loaded along with rehydration buffer or can be added after rehydration of strip. Larger size protein molecules cannot enter into gel matrix directly, and applied voltage in the range of 30 to 100 volts can pull the protein molecules into matrix. Smaller molecules enter into the matrix by absorption. Dear students, by now I have covered theoretical aspects of the isoelectric focusing technique, and you must be very clear that proteins differing in their isoelectric point can easily be separated by isoelectric technique. If you see the resolving power of this particular technique, that proteins, if they differ in phosphorylation or in glycosylation, these two proteins can be separated by isoelectric focusing technique. Now my focus will be on the practical aspect of the isoelectric focusing technique, in which we will discuss that how the sample can be prepared, how the gel can be casted, and how these can be stained, and also we will discuss whether this technique can be used in conjunction with HDS-based gel electrophoresi. That is what is referred as the two-dimensional gel electrophoresi. We will see that more applications of the isoelectric focusing technique in two-dimensional gel electrophoresi. Sample preparation isoelectric focusing is critical process. Protein sample should be clear and should be clean. It should be free from salts unless otherwise exclusively needed for solubilization. It should be free from buffer components and ionic detergents. Ampholytes contribute to ionic strength and can help solubilization of protein. proteins from cell or tissue are required to be extracted in presence of proteolytic inhibitors, especially when detergents are used for extraction of membrane proteins. Purified proteins can be solubilized in water or 7 to 8 molar urea. Urea solution should be free from cyanate, which is present in old urea solutions. Complete denaturation of proteins requires treatment of sample with thiol reducing agent. Urea at this high concentration can enhance viscosity and reduce effective size of water channels. Membrane proteins may require detergent for solubilization. Non-ionic detergent such as CHEPS, NP40, Triton X100 can be used, but these should be free from contamination of ionic detergent. Few microliters of sample in the range of 1 to 10 microliter containing buffer can be tolerated. Results are better in deionized water, 2% ampholytes and 1% glycine. Salt or buffer in sample can be removed by dialysis or gel filtration. Once the gel is polymerized, the glass plates are separated in such a way that gel sticks to one of the glass sheets. Electrode wicks, which are thick wetted filter paper, lay along the length of each side of the gel. Anode chamber is filled with phosphoric acid, while cathode chamber is filled with sodium hydroxide. Then potential is applied. This will result in formation of pH gradient between anode and cathode. Then potential is switched off. Sample can be loaded on a small piece of filter paper, which can be subsequently placed on gel and slightly pushed. It is preferred to load sample about 1 centimeter away from each end. Potential is again applied for about 30 minutes for sample migration from paper to gel. At this stage, paper is removed and potential is again applied for 2 to 3 hours. High potential is required for isoelectric focussing and voltage of 2500 volt can be applied. As a consequence, considerable heat is generated. Thus, gel must be cooled to 10 degree centigrade for avoiding band diffusion. When electrophoresis is completed, the gel is fixed in 10% trichloric acetic acid. This immobilizes the protein in gel and also removes ampholytes from gel. Presence of ampholytes in gel results in background staining when gel is stained with commercial blue. The gel is also de-stained as in case of HDS space. Isoelectric focussing can also be used for preparative applications. In a vertical column, add mixture of Cephedex G75 as gel matrix, ampholytes and sucrose to create gradient in column. Once separation is completed, elute out the sample for further process. Proteins differing in mass and charge can be separated both by HDS phase and isoelectric focussing. However, proteins of the same or very similar mass and differing in charge cannot be separated by HDS phase. These proteins can be separated in isoelectric focussing. When proteins only differ in phosphorylation, such proteins will have a small difference in their charge or pi values, and these can be easily resolved in isoelectric focussing. Thus, the technique can be used for identification of microheterogeneity in protein. Isoenzymes differing in their isoelectric point can be identified. This can be achieved in two ways. The gel can be exposed with substrate, which on action of enzyme gives color reaction. For this purpose, gel can be dipped in substrate solution for few minutes and appearance of band is then noted. Alternately, agar gel can be layered over isoelectric focusing gel for transfer of protein to agarose gel, and then enzyme is stained for its activity. Other application of isoelectric focusing is in determination of isoelectric points of proteins. Isoelectric focusing not only separates proteins based on their isoelectric point, the technique can also be used for determination of isoelectric point of new protein. The procedure requires running of protein standards, which are the mixture of highly purified proteins whose isoelectric points are known. These standards are run in a different lane but on the same gel on which the unknown protein sample is separated. After completion of run, gel is stained for protein. The distance of each band corresponding to different standard proteins from either end of the gel is noted. Also, distance of unknown protein is similarly noted. These values are then plotted on graph. Distance and pi values are recorded on different axes. From standard curve, one can calculate the value of pi's corresponding to the distance of unknown protein. Other application of isoelectric focusing is in two-dimensional electroforces. This is combination of isoelectric focusing and HDS space techniques. In isoelectric focusing, samples are separated on the basis of isoelectric point, whereas in HDS space, separation of protein is based on difference in molecular mass. The resolution power of two-dimensional electroforces is far better than either of isoelectric focusing or HDS space. In this technique, proteins similar in mass but differing in charge can be separated. Also, proteins similar in charge but differing in mass can also be separated. The technique is performed by first separating protein in isoelectric focusing and then separating the proteins on sodium rhodocyl sulfate polyacrylamide gel electroforces. This technique is of choice when sample contains large number of proteins. New proteins in biological sample can be identified by comparing band pattern with normal sample. Isoelectric focusing is performed either on IPG strips or on gel filled in narrow glass tube of 1 to 2 millimeter internal diameter. This is done in presence of 8 molar urea, Ampholytes and non-ionic detergent. After isoelectric focusing, gel from the tube is collected and then equilibrated in buffer containing HDS. This gel is then placed over HDS space gel and ensured to make a good contact with stacking gel. Contact can be further ensured by pouring some molten agar. When agarose gel is set, HDS phase is initiated. On completion of run, gel is taken out and stained for protein. In 2G gel electroforces, proteins are resolved in 2 dimensions and therefore large number of proteins can be identified from a single run. The technique is routinely used in proteomic labs. Dear students, if I want to summarize the technique of isoelectric focusing, I made it very clear that this technique has high resolution power, and if two proteins differ in their isoelectric point, these can be separated. Proteins which differ in phosphorylation, proteins which differ in glycosylation, that is most of the situation with the isoenzymes. So these can be easily resolved in isoelectric focusing technique. Further, I want to make it very clear that if we know the sequence of the protein, then using the bioinformative tools, you can calculate the isoelectric point. So it is not essential that you have to run the isoelectric focusing for determination of the isoelectric point of the unknown proteins. If protein is glycosylated or protein is phosphorylated or modified in other way, then it is essential that we can determine the isoelectric point by using the isoelectric focusing technique. But we see the better use of the isoelectric focusing in conjunction with SDS space, where the proteins can be separated based on their mass, based on the charge, and it is possible to separate even hundreds of proteins using the two-dimensional electric forces. So most of the applications of the isoelectric focusing are in conjunction with the sodium dioxide sulfate polyacrylamide gel electric forces. If you have any bench problems, you can always contact me. I am always available to help you out if you have faced any problems on the bench, and I welcome for this listening to this particular lecture for helping you to enriching your knowledge in the isoelectric focusing techniques in particular.